And our next speaker is Eran, and I think you get the award for the longest duration flight to come to this conference. So thank you very, very much. Weizmann Institute, please.
So, good morning. I'm very happy to be here. I noticed yesterday that everyone has a very cool spacecraft photo. So I just wanted to bring up the point that I come from a country smaller than the size of New Jersey, but we do have our space program. We had an astronaut that unfortunately lost his life in the Columbia disaster. And a few months ago, we became the 7th country to have an orbiting unmanned spacecraft around the moon, and almost became the 4th country to land the spacecraft on the moon. It crashed. We don't believe in backup systems in my country. So to switch gears into the microbiome, which needs no introduction after the previous two talks, but one point that I will be bringing up is that the deeper we sequence humans with the microbiome, and it doesn't matter if it's the skin, the gut, or any other microbiome, we come to realize that each of us carries a very unique signature of functions and compositions of their microbiome. And as you will see, we like this trait and we harness it in order to better understand human physiology and disease. And in my group at the Weizmann Institute of Science, we regard the microbiome as a signaling hub that integrates into it many signals that come from the host and from the environment, and receives them to the host in a communication channel which still is poorly understood, but we see our mission in life in decoding the alphabet that makes this language by which we communicate with our microbes to understand how physiology runs and what happens when this language is perturbed that leads to a development of multifactorial disease. And of the many different factors that impact the microbiome, I thought it would be most relevant to briefly talk about the number one influencer of the microbiome by far, which is diet. So of all the different things,
Can you publish in some other journals?
I'll do my best, Jack. M systems. So let's talk a little bit about diet and how it influences the microbiome and how we can learn from these interactions between diet and the microbiome on human health and the propensity for human disease. And the groups of diseases that we've been focusing on are no strange to this crowd for sure, and constitute probably the worst epidemic in our species history, which is the obesity, type 2 diabetes, fatty liver, and metabolic syndrome epidemic. You can see the numbers in the US. They are true in almost any other region in the world, including now India and China, including children. And we really have no viable solution for this tremendous epidemic in around a century. And in trying to understand how the dietary changes, which we believe are a major factor that contributes to this epidemic, mediate changes to the microbiome and through the microbiome to human metabolism and immunity, we can kind of conceptualize these relationships as diet impacting the microbiome in 3 different ways. First, the composition of diet, the composition of the foods that we eat has a potential impact on the microbiome. But an equally interesting impact of diet is the timing of diet, and I'll show you one example of our own work relating to this. And long-term dietary patterns have been recently suggested to also bear a very important impact on the microbiome. And I'll just share with you a couple of vignettes of our own work exemplifying each of these subtopics. So we'll start with the composition of the microbiome, and we always like to start our project with a very naive question. So we asked ourselves, can we gather some new information on how the composition or the types of diets that we eat impact our microbiome, and through our microbiome impact our metabolic health. So I started reading all the very boring literature that forms the basis of everything that every one of you are doing in your daily lives when you try to come up with a so-called healthy diet. One really surprising, at least for me, point that came up was that everything that we do is based in one way or another on one of several grading systems that give different numbers to different foods or food components. So for example, calories are such grading system that gives different numbers to different foods, and what we basically are all trying to do is to mix and match these numbers to come up with a diet which we believe is healthy for us. Even more commonly used than calories is a system called the glycemic index. And take my word for it, any diet that you've ever tried, going to your dietitian, family physician, buying a book at the airport, is based in one way or another on this glycemic index system. So what is the glycemic index score? So it's based on very small human trials that were conducted in the 1970s. These trials usually included 10 human volunteers, and these volunteers were kindly asked to eat an identical food, and then their blood sugar levels were measured for 2 hours after they ate this identical food. So for example, if you would take 10 human individuals and would give them an identical piece of celery and measure their blood sugar levels for 2 hours, you would get this sugar spike like in this black graph. The average rise would be given a number, and that number is the glycemic index of celery. Now, if you would take the same group of individuals, and now you kindly ask them to eat a good piece of chocolate cake, their average blood sugar rise would be the blue graph, and this higher number would be given a number, and this would be the glycemic index of chocolate cake. Now, if you open your smartphone, you would find endless tables that give you a glycemic index to any food or food component on Earth. And again, take my word for it, anything you've ever done with your own diet or with your patient's diet or with your astronaut's diet is based in one way or another on this glycemic index system. Now here comes the big problem. When we repeated this experiment, but not with 10 human volunteers, but rather with 1,000 human volunteers, and we measured the responses to identical foods, when we gave, for example, test foods that we generated, such as 50 grams of glucose or an identical piece of white bread to all of these 1,000 individuals, and measured their blood sugar levels on the left or left panel, the average response of these 1,000 people to that particular food was exactly the glycemic index of that food. So the glycemic index is absolutely true, and it's absolutely accurate. But when we looked at the individualized level of the 1,000 individuals, some individuals spiked their blood sugar levels to diabetic levels, while others didn't care at all. They could eat or ingest 50 grams of glucose or white bread and not even change a bit of their glycemic responses. This was true for the test foods on the left. This was also true for hundreds, now thousands, of real-life foods that we've measured in a study that I'll share with you. And in all of them, the variability in human responses to identical food was unimaginable. And this already intuitively told us, and should tell you, that this one-size-fits-all dietary approach, which we were hunting for decades, cannot be true by definition, because if your glycemic index or glycemic response to a given food is opposite than mine, then the same diet cannot be good for you and for me. And this gave rise to a very ambitious project, which we called the Personalized Nutrition Project, in which we kindly asked these 1,000 people to give us a week of their life. And during this week, we've collected an unprecedented amount of person-specific information, including, given our interest, a very deep dive into the gut microbiome by both 16S and Shotgun, many blood tests, many questionnaires that they fill out with regard to their lifestyle, medical illnesses, and so on and so forth, and anthropometric measurements. We developed a smartphone app for this project that enabled us during a week of follow-up to learn everything about each of these individuals, when they were going to sleep, when they were waking up, how much they were exercising, what and how much they were eating, and so on and so forth. We connected each of them to a continuous glucose monitor, which measures their blood sugar levels every 5 minutes for an entire week. So in contrast to the 1970 studies that I've shown you before, in which 6 blood sugar measurements were taken, here we had 2,500 blood sugar measurements taken from 1,000 individuals. And the most important and smart thing happened after the week of follow-up, when a very smart group of computational graduate students took this huge amount of person-specific data from each of the volunteers and used AI and machine learning techniques in order to devise a personalized algorithm that is aimed at predicting a person's glycemic response to any given food, including foods that they haven't consumed during the week of follow-up. And to make a very, very long story short, this is the gist of the results. On the left, you can see what we calculated to be the predictability of what happens when we go to our family physician or a dietitian and ask to reduce our sugar blood levels. And there is some predictability to this approach because we're usually advised to eat a low carbohydrate diet, which has some merit to it. And this is the 0.38. But when we use our person-specific, science-based AI approach, we dramatically increase our capability of predicting a person's response to any given food to 0.68. And as is customary in these drills, after you train a machine learning algorithm, you need to freeze it and to apply it to a blinded new cohort of individuals, another 100 individuals we've used, and we could reach the same exact, very high level of predictability. So we were very happy with ourselves, but it was now time to put ourselves to the real life test. So we recruited a new batch of, or a new group of human volunteers. This time they were all pre-diabetic, so they already suffered from perhaps the most common feature of the metabolic syndrome. In the US population, it is estimated to include 40% of the adult population. As a physician, I can tell you that we have very little to offer them. We tell them to exercise more, to eat less. They never do. And they have an 80% chance of developing type 2 diabetes within the next decade of their life. So we took these individuals, we put them through our process, but this time we kindly asked the algorithm to generate a group of good personalized diets and bad personalized diet per each individual. And we asked him that these two types of diets would be isocaloric, so we won't blame calories. And then we asked our volunteers to eat their personalized good diet and then their personalized bad diets a week at a time, while we really extensively continue to measure them, including measuring the microbiome every single day of the intervention. And of course, the diets were different between different individuals. They're personalized, and they were super counterintuitive. So a person could have ice cream as a good component of his or her diet. Actually, in 72% of the population, this was the case. A person could have a certain vegetable as a bad component of his or her diet. So everything we kind of think we know, we don't know until we measure it. And the results were that even a week of good intervention normalized the blood sugar levels in these individuals that are pre-diabetic after just 1 week, while the bad personalized nutrition spiked them even further. We are now finishing up a really large-scale follow-up study in which we do the same drill for a whole year, a long-term study, and we compare it head-to-head with the gold standard American Diabetes Association diet that we already know doesn't work. And we'll see what the results are. And the reason I'm telling this to you in a microbiome session or a microbiome meeting is that after we finished all of this algorithm generation and follow-up, we can now go back into the algorithm and check what it shows in an unbiased way in order to form an accurate prediction. And what we found was that of all the thousands of features that were fed into the algorithm, by far the ones related to the microbiome formed the largest group. So the microbiome seems to be a very important component in our ability to predict a personalized dietary response. We're now studying and following up the different features, and actually we can now reach the same level of predictability by just taking people's microbiome plus a list of clinical features, even without measuring their blood sugar levels. This told us that everything is personalized, and then we followed up in a number of studies focusing more deeply on other personalized nutritional features that came up in this large-scale study. For example, one study discovered that one of the most personalized foods are non-caloric artificial sweeteners, which are taken by millions of people with an aim of reducing their blood sugar. And what we found was that some people with some microbiomes actually feature a counterintuitive increase in their blood sugar levels and their glycemic responses when they're exposed to these supposedly inert substances that are not inert to the microbiome. Another example that we've explored is bread, because in my region of the world, bread includes around 30% of the caloric intake of all individuals. So we performed this crossover study in which we took a group of individuals, gave half of them the white industrial bread, which you all would think is not good. The other half got the very best bread in the world, handmade by the very best chef in Tel Aviv, French yeast, cost me tons of money. And they ate these two breads, and then they crossed over. The group that ate one bread ate the other bread. We extensively monitored them, and to make a long story short, we found that there is no good bread because half the people spiked on one bread, half the people spiked on the other bread, and we could use the starting microbiome in order to predict very accurately who would spike on which bread. And a few months ago, we even found that probiotics, which are a highly controversial topic, are also personalized, and people's colonization of 11 types of probiotics are personalized to the fullest, and we can predict based on starting microbiomes who would colonize probiotics and who would just pass them from one end to the other. So these were kind of examples of how food composition impacts the microbiome, and through the microbiome may impact human physiology and disease. But as I said at the beginning, we also found, very surprisingly, that dietary timing and the timing of feeding has profound effects on the microbiome and the human body. And in this project, we again started with a very naive question. We have a great interest in circadian activity. And the circadian field, which I'm not part of, has been blooming in the last 20 years, and we discovered that we have one central clock in our hypothalamus, but we also have a clock in each one of our cells and organs. So we have millions of clocks in our body ticking amazingly in a highly coordinated manner and dictating almost every physiological function that we have. And now we regard the microbiome as a sort of neglected organ. So we ask ourselves, does the microbiome, like any other organ or cellular system, has a diurnal activity? Does it change its behavior during a 24-hour cycle? So this started a very painful set of experiments in which my students sequenced mice and humans along different times of the day in consecutive 24-hour cycles. We use very stringent statistical tools in order to make sure that we are accurately looking at something that is continuously diurnal in its behavior composition, and we found, very surprisingly, that the gut microbiome is highly diurnal in its composition and function. So you can see here an example of one of these analyses. Every dot below this dotted line featured a very stringently statistically significant diurnal activity. You can see an example of one of the genera going up during the night, down during the day, up during the night, down during the day. You can continue forever, and it will be like this. With respect to function, using shotgun, you can see here an example of a few tens of microbial functions in the gut, going up during the day, down during the night, up during the day, down during the night, and so on and so forth. And this was very surprising to us, because if you think about it, the microbes live completely in the dark, so how the hell do they know that it's day and night and change their behavior? And this led to 3 years of research, and the answer at the end was that the major factor which tells our microbes which time of the day it is and tells them to change their behavior is the timing of our feeding. So for example, if we took groups of mice and fed them only during the night or only during the day and measured their microbiome across 48 hours, you can see that we can phase shift the behavior of the microbes by 12 hours. So the feeding behavior is the major regulator of this microbial behavior. Now this is all interesting scientifically, but is it important physiologically? And I think that it is important because for millions of years, our body learned how to behave in a perfectly circadian alignment. So we woke up in the morning, we worked, and were active during the day, and then we went to sleep during the night. But in a glimpse of evolution, in 150 years of industrial revolution, all of a sudden we beat this evolution. All of a sudden we invented light bulbs and electricity. And now we have millions of people engaged in shift work. We generated planes. Now we can travel across time zones. And together with this chronic disturbance of circadian activity, there is a huge amount of literature showing that these chronically circadian disturbed individuals have a huge risk of developing obesity, type 2 diabetes, and even cancer. But we never knew what the missing link was. And we asked ourselves whether this microbial circadian activity could contribute to this risk behavior. So in order to answer this question, we performed a series of experiments in mice in which we induced a very severe state of circadian disturbance. If these mice were humans, they would be flown from Tel Aviv to Tokyo and back every 3 days, 7 times in a row. So this would be one big jet lag, just like the one I am experiencing right now. And in humans, we performed a series of experiments that made me very popular at the Weizmann Institute at the time, because I took a group of students and I flew them all over the world, and all I asked them was to collect poop and to bring it to me so we can see what happens during, before, and after jet lag. And the answer was that the microbiome really changes really dramatically during circadian disturbance, but to prove that causality, which is one of our hallmarks at the lab, we took the jet lag microbiome or the non-jet lag microbiome from mice or from humans, and we transferred it into sterile germ-free mice. They don't have any microbiome of their own. And what we could see was that the transfer of a jet-lagged microbiome into these mice was enough to induce a state of severe obesity and type 2 diabetes, both when mice or human microbiomes were transferred, suggesting that perhaps these microbiomes, diurnal disturbances, are at least partially responsible for the ensuing risk behavior in these populations. And we're now finishing up very large-scale human studies that are proving the same. And the last vignette that I'm going to share with you relates to long-term dietary patterns, which we found to also dramatically impact the microbiome and human metabolism through the microbiome. And the disease we chose to focus on is called recurrent obesity, or yo-yo obesity. I'm sure many of you are aware of it, because if you look at the numbers, 80% of all overweight individuals in the world follow this pattern of obesity. And this pattern basically consists of a person that becomes obese for any reason, and then goes on one of hundreds of diets and loses back his or her weight successfully. And this is always the case in the short-term period, but within 12 months of successfully dieting, 80% of individuals go on to jump back and become obese, and usually a little bit more obese than they started off. And from cycle to cycle, obesity develops. This is yo-yo obesity, accounting for 80% of all obesity in the world. Now, if we're starting to scrape the surface in understanding obesity, we really know almost nothing about the causes of this recurrent obesity. So we wanted to contribute to this field, and we developed 3 different animal models of recurrent obesity. For the sake of time, I'll just show you one of the 3. It's a very simple 4-mouse experimental setup. You can see that one control group of mice in black just eat normal chow, so they gain very little weight. Another control group of mice in blue are eating now a high-fat, high-sucrose diet, and as expected, they gain a lot of weight. But the important groups are the groups in the middle. For example, the red group starts by eating the high-fat diet, gains weight just like the blue group, and then we switch it to normal chow, so it diets back to black levels, and then we give it another round and another round. And in each round, we add another group of yo-yo dieting mice. So if you look at the 3rd cycle of obesity, you can see that although all mice started from the same exact weight, the mice that are in their 3rd cycle have an exaggerated weight regain, these red mice, as compared to the green mice that are in their 2nd cycle, or the orange mice that are in their 1st cycle, which is a great representation of what happens to humans with this yo-yo obesity phenotype. And this is not just true for obesity, it's true for any other metabolic feature that we measured, which is exacerbated from cycle to cycle of obesity. Now, the billion-dollar question is, what drives all of this? What in the mouse remembers that it was once obese and predisposed it to an exaggerated weight regain in the next round? So we focus on this period after a successful dieting, which we called the nadir period. In this period, the mice are not different in weight from mice that were never obese. And we asked what physiological feature in the body of a mouse at that period is disturbed as though it was still obese. So we measured really hundreds of different endocrine and immune and you name it features, and everything seemed to normalize after a successful diet. Everything but one factor, the gut microbiome. When we measure the gut microbiome, after a successful diet, mice in red assumed this intermediate configuration. So the microbiome was somewhere between the obese microbiome and the lean microbiome, but it never really returned to normal. And this was true in every level of microbiome analysis that we've measured, in composition, in metagenomic function, or even in the pathway level of metagenomic function. Now, this was an interesting observation, but it tells you nothing about whether this microbiome drives exaggerated obesity. And in order to prove causality, we use many different methods. This is what we do for a living in my lab. And for the sake of time, I'll just show you one killer experiment, again, involving these sterile germ-free mice. And we took the nadir post-successful dieting microbiome, or microbiome from mice that were never obese, and we transferred them into germ-free mice that were now induced with high-fat diet. And you can see that mice that received this memory microbiome in orange developed more obesity and more glucose intolerance or diabetes as compared to mice that received the never obese microbiome, saying that, or suggesting that there is some information stored in this post-successful dieting microbiome that predisposes to an exaggerated weight regain in the next cycle. So we wanted to dive a little deeper. We developed computational pipelines incorporating our genomic and our metabolomic data sets. We use metabolomics, which characterized by mass spec the tens of thousands of small molecules that are associated with different microbiomes. And in this case, we were very lucky because we found one genomic pathway encoding for 2 isoflavonoid metabolites that were suppressed in this memory post-dieting microbiome. And this became our main suspects, and in order to prove that this was the case, we replenished the missing metabolites into this post-dieting microbiome in mice. And when we did this and re-challenged the mice for a second obesity bout, they no longer developed this exaggerated weight regain. So it was the fact that these 2 molecules was missing in their microbiome that was not able to properly generate them, that was driving, at least in mice, this exaggerated weight regain. And then we probed even further, and we found that these 2 molecules actually swim into the sterile host, where they impact the adipose tissue in the sterile host to generate more heat and less fat generation. So when they're present, they tilt the balance towards heat generation rather than fat accumulation. And when they're missing, they do the opposite, and exaggerated weight regain ensues. Now, we published this in Nature, and 2 weeks later came this New England Journal paper following a group of recurrent obesity individuals, I think 10,000 of them for 10 years. This was not a microbiome paper, but what was important about it is that it showed that recurrent obesity is not just very common, but it is an independent risk factor for the development of type 2 diabetes, of cardiovascular complications, of all mortality causes. So it's a very important feature of obesity, and this led us to start a clinical trial in which we try to implement all the technologies that we've developed for our mouse project to see whether we can identify signatures in recurrent obesity human individuals that may contribute to their phenotype. So this is ongoing. And just to finish, I thought to give you kind of my 2 cents on where I think we are as a field in terms of the utilization or the translation of all the data that you've heard and you will be hearing in this meeting and in others towards human health. So, in 2018, to be very modest, we're still talking therapeutically about 3 avenues of microbiome research. One is prebiotics, which is the consumption of certain compounds in diet that are presumed to impact the microbiome. FMT, which is fecal microbiome transplantation, which is a brute force way to replace the microbiome. Very successful in C. diff infection, less successful in other diseases, and probiotics. And all of these have very many limitations and controversies associated with them. But I think that the field is slowly moving towards a more data-driven, science-driven individualization of microbiome data in exploiting it towards human health. I've shown you one example with personalized nutrition. There is a lot of activity going on with moving from these yogurt-associated probiotics towards next-generation probiotics that are personalized, data-driven, harnessed to the individual. So that's very interesting. There is a new way of treating through the microbiome, which we call postbiotics, which is identifying small molecules, such as the example I gave you with recurrent obesity, that are missing in some disease contexts and giving them and bypassing this entire inter-individual variability of the microbes themselves. I've not talked about ways that we're developing for targeted elimination of microbes. So let's say you have a microbial signature that is associated with a given disease. We right now have 0 ways to take them out of the microbiome without killing the entire microbiome. Antibiotics are for sure not a specific manner. So there's lots of activity there. We're dealing with phage-related technologies that we're developing for this task, and others are developing other techniques. And of course, we should not forget the host. There are very interesting therapeutic targets that would probably be identified in the next few years coming from the host in these interactions, and they potentially could also be exploited. And what we hope is that in a few years, all of these tools will be on the shelf, and different clinicians or different purposes could be using them as part of the therapeutic armament. And I will finish by thanking the people who have been doing all of this work from my lab, our great collaborators, including Eran, a mathematician from the Weizmann, who's been engaged with us in many of these studies, and other clinical and scientific collaborations, the funding agencies that are making our work possible, and I'll stop here. Thanks.